This Week in Microbiology is brought to you by the American Society for Microbiology at asm.org/TWiM. This is TWiM, This Week in Microbiology, episode 204, recorded on July 9th, 2019. I'm Vincent Racaniello, and you are listening to the podcast that explores unseen life on Earth. Joining me today in studio here at Columbia University from the Vagelos College of Physicians and Surgeons of Columbia University, Nicholas Arpaia. Welcome, Nick.
Hi.
My colleague. You're my colleague here in the department, right?
Yes.
I've known you for a few years.
Yeah.
Should I call you Nicholas or Nick?
Oh, you can call me Nick. I mean, this is like, it's like, we're colleagues.
Yeah, we are.
Yeah.
We are colleagues. Also from Columbia University, Tal Danino. Welcome to TWiM.
Hi. Thanks for having me.
So you're at the Morningside campus, is that right?
Yeah, that's right. 120th Street.
Okay, so we have to tell everyone that I have been here at the medical school since 1982, guys. Long time.
Wow.
Were you born?
No.
1982, yeah.
No,
You weren't born?
Not. You were not.
You were.
Yeah.
No, you were not.
Well, it's the last day of 1982, so no.
I've been here a long time at the medical school. And there's, of course, a campus the medical school is at 168th Street on the west side of Manhattan. And we have a campus at 116th Street called the Morningside campus. And that's where Tal is, right?
There's grass there.
There is grass.
It's nice.
Yeah. Here we have concrete.
Yeah. Yeah.
And concrete and concrete.
But I never have I never have a problem going to visit Tal because I get to walk on grass. How
Yeah, it's
often
very
does
nice
that
down
happen in the city?
I like it down there. I teach a course there in the spring every year. I really like going down there. It's fun. All right. So we're all at Columbia University essentially, and we're recording in studio today, a little special episode because these individuals have published a really cool paper that I want to talk about. And so before we get to that, let me just remind everyone, if you like our work on these podcasts, consider supporting us financially. Go to microbe.tv/contribute. I'll have more about that later. I want to start by figuring out where you guys have come from. Remarkably, Nick, I barely know your history.
Oh.
Where are you from originally?
I was born in Mount Vernon, New York.
It's right up here.
Yeah,
right
here.
Oh, it didn't fall far from the tree. And you went to high school in Mount Vernon?
No, I went to high school in Thornwood.
Okay.
So by north of White Plains.
Okay. Where'd you go to college?
SUNY Geneseo.
Wow.
Yeah.
Upstate New York.
Uh-huh.
They used to have a beer called Geneseo.
Geneseo, yeah.
Right?
That's a good beer.
Because I went to Cornell and I remember
yeah.
That horrible beer. I think it was horrible, wasn't it?
I don't know. I mean, it was college, right? Was there any bad beer?
Yeah, right.
Yeah.
So you were a science major at SUNY?
I was a biochemistry major, focused more on chemistry.
Uh-huh.
So I took more chem electives than I did bio electives. But, we covered everything.
When did you get interested in science? Remember?
So my mom worked at Mount Vernon Hospital. She was a secretary in the laboratory for a long time. And she would take me to work sometimes, and I would get to hang out with the pathologist.
Oh, cool.
And the microbiologist. So I kind of got interested then.
After SUNY, where did you go?
So 2 weeks after graduating, I went straight to Berkeley, UC Berkeley, to start my graduate work.
Wow.
Yeah.
Pretty nice place, huh?
Yeah, it's so beautiful.
It's beautiful there. Who'd you work with?
So I did a summer rotation that summer with Mark Schlissel, who's now president of
Michigan.
Yeah, Michigan. But I did my graduate work and my thesis work with Greg Barton studying toll-like receptors, so focusing on the innate immune system.
I think we interviewed Schlissel for chair of this department ages ago.
I've heard that.
Yeah.
I wasn't around then, but
yeah, sure. But I was at an event at Michigan with Schlissel, and I took a selfie, and he thought that was really funny. It was really, how many university presidents get to do that? Okay, after Berkeley...
then I came back to New York, where I'm from. I joined Sasha Rudensky's lab at Memorial Sloan Kettering.
Mm-hmm. So that's the part of you I know. And you continued working in immunology, right?
Yeah. So I transitioned from studying innate immunity to adaptive immunity, really focusing on regulatory T cells, which suppress responses by the adaptive immune system.
Got
so
it.
they're a unique subset of T cells.
And then you came here. This was your first faculty position. What year did you come here?
Hopefully my last, right?
Yeah. Well, it depends. You might want to go to Harvard or,
No,
Stanford or Berkeley.
I would be happy to remain colleagues here with you guys.
You would?
Permanently. Yeah.
Okay.
It's great.
What year did you come here?
I started here in September of 2016.
Okay. You're so young. Only 3 years. Okay. Tal, where are you from?
Originally from Los Angeles.
Wow. Grew up high school and all that there?
High school was there, yeah.
And where'd you go to college?
College was also there at UCLA.
Nice. And you were a science major?
Yeah, I was mostly studying physics and math. So
Mhm.
more on the quantitative physical sciences.
Right. And how did you get interested in science? You remember?
Interested in science. I think I was always interested in, computers and technology. And then I think the science came a little bit later. It was actually mostly through chemistry and doing experiments around the house, trying to, make little fireworks and things like that. And then that sort of led to an interest in biology and living things and how they work and using physical sciences to do that.
I know quite a few people at UCLA, but they're life scientists. You probably wouldn't know them.
Mm.
And you probably know, Owen Whitty is the famous one. He was an immunologist from the Baltimore lab, discovered ABL as a tyrosine kinase. Don't know that name? Okay, that's
I
fine.
interviewed in the micro department.
Did you interview with Ashim Dasgupta? Remember that name? No. He's a virologist. And Arnie Burke is at UCLA. He and Phil Sharp did splicing, but Arnie didn't get a Nobel Prize, unfortunately, Only Phil Sharp got that. What did you do after UCLA?
Then I went to UCSD for a PhD in bioengineering. And there I worked with Jeff Hasty, and we were basically building genetic circuits in bacteria and just fundamental programming of bacteria and how do you model and do that.
Mm-hmm. Okay. I was just in San Diego for a day last week. It was nice weather.
It's nice there.
Always is. All right. So that was your PhD. And where'd you go for a postdoc?
Then I went to MIT and I was in Sangeeta Bhatia's lab there, which is part of this Koch Institute for Cancer Research. So it's an interdisciplinary place that mixes engineers and cancer biologists. So that's where I started working on cancer research.
So I spent 3 years at MIT from 79 to 82, just before I came here. I was a postdoc with David Baltimore.
Everybody, wow.
A lot of people.
Yeah, especially in this department.
So Steve Goff, of course. Fred Alt, who used to be here, was a postdoc. Shankar was a postdoc. That's right. That was after me, though. But Fred Alt, you know that name, right?
Yes, of course.
He was here for a while. Then he went to Harvard and has done so well. But he was a postdoc with David. And who else? Is there anyone else? Oh, yeah. But Richard Mann, the fly guy, this building. I don't know him. He was from David's lab. Anyway. And then you came to Columbia after that, that postdoc at MIT. What year did you come?
So that was January 2016.
Same year you guys both came.
Yeah. Yeah, same year. Did you go to public high school?
I went to a public high school.
Yeah, so we were public school educated straight through to our postdoc.
Yeah.
Interesting. I'm curious how this collaboration began. Did you go to a seminar? How did it work?
I think it was through a student who joined our lab, who was the first author on this paper, Shreyan. And I think he was either rotating or had already joined the paper. And we were engineering these bacteria to deliver drugs. And so that started to get into Nick's territory, into what kind of drugs we should make to stimulate the immune system. So I think he actually connected us, or he told me to reach out to Nick.
Yeah, Tal reached out and, you see another assistant professor email you and you're like, yeah, of course.
Yeah, it's great.
So we met and we talked about it. We set up, we had a couple of meetings, I guess. And our student, Shreyan, was initially in Tal's lab, directly in Tal's lab. But then as the project developed, we were like, okay, well, we should take him on as a joint student. And then it became really focused on, understanding certain immune targets, developing several of them. And then really a true collaboration, where we talk about the different circuits and, come up with different targets and try to modulate the immune system that way.
So you're the immunologist on this, right?
Yeah.
And Tal, you would not consider yourself an immunologist. You're a circuit person.
I'm the engineer, yeah.
You're an engineer, but
medical
you
engineer.
probably knew some tumor biology.
I knew some cancer, very little immuno stuff.
But you've become... He can
I've
speak immunology pretty well.
Yes.
When you collaborate, you have to learn, right? And that's good. It's the way it works. So this is a paper that just came out last week, I guess. And so I heard this part of this story. Jeff Hasty came last year and gave a talk, and I said, wow, this is amazing stuff. And we'll talk about this idea of programming bacteria to do things. And his slides were just amazing.
Yeah, Jeff
beautiful.
Jeff gave a great talk.
And I said, I had to get this guy on TWiM sometime. And then I guess you were in the audience, Tal. And I said, I don't need Jeff. I'll get these guys to. And I heard about the collaboration. And I approached you a long time ago, but you said, let's wait for the paper.
Yeah, I was still a little nervous. You don't want to jinx it when you feel like the paper might come out. So
no worries. It just came out in Nature Medicine. It's called Programmable Bacteria-Induced Durable Tumor Regression and Systemic Antitumor Immunity. And so the first author is Shreyan, and he's in your lab, Tal, as a PhD student?
Well, he's a joint student, yeah.
Oh, that's cool. That's very cool. So tell me the roles of the other people. We have Sam Castro. Is that shared?
He was actually an undergrad in our lab.
A Columbia undergrad. That's cool.
Yeah.
Courtney Coker.
She is a research staff in our lab.
Okay. Taylor Hinchcliffe.
And Taylor was also a research assistant. Yeah. So all three of them were really responsible for working on the animal studies.
Right. And then we have Nick and Tal, the two anchor. We would call them the corresponding authors.
Mm-hmm.
So this is really cool. I was thinking about how to attack this. I think let's first look at engineering the bacteria. You have this very cool thing, this SLC, synchronized lysis circuit that you built in E. coli, right? And you must have worked on this in Jeff Hasty's lab, right?
Yeah, exactly. So I think it probably goes back a while to a time when I was in Jeff Hasty's lab and building circuits that involved quorum sensing. And then on top of this quorum sensing, some other grad students in the lab, one Arthur Prindle and then Omar, who actually made this lysis circuit, built this quorum sensing circuit plus another lysis gene. So that when the bacteria hit a critical mass or they hit a quorum, then it will actually cause them to self-destruct. And the purpose of the self-destruction, I think some people have thought about that idea before, just to limit the growth of bacteria. But in this instance, the specific purpose was to get them to lyse and actually release a therapeutic that they encode genetically.
So that was the idea from the beginning, to deliver something by lysing.
Yeah, exactly.
And I guess also all the immunostimulatory products released from bacteria would help the immune response too, right?
I mean, that's like when I saw the SLC, or they talked to me about it the first time, it was like, well, we have signal 1 and signal 2 we can modulate. So it's exciting.
So can you explain how this works, this synchronized lysis circuit? It's E. coli that you engineer to do this, right?
Yeah, E. coli or salmonella. So the original 2016 paper was using salmonella. And that was basically proof of concept, more of the circuitry and some preliminary animal results. But I guess, there's sort of two aspects. One is the bacteria make it to the tumor and they will selectively grow in tumors.
Mm-hmm.
So that's, a really interesting fact on its own. And they grow in these tumors selectively because there's not a lot of immune surveillance. So that's the sort of the bacteria cancer aspect to it. But then specifically this circuit, the way it works is the bacteria grow and they're making this small molecule AHL, which is a quorum sensing molecule, and it can freely diffuse between bacteria. And so once there are enough of them making enough of this molecule, then it hits a critical threshold that turns on this promoter.
Mm-hmm.
And then this promoter would be driving either a fluorescent reporter just to allow us to visualize the circuit, or, for instance, the lysis gene, which would cause them to self-destruct.
It's a phage lysis gene?
It's a phage lysis gene, yeah.
We have to plug the viruses here.
Yeah, it's a
This is TWiM.
X174.
Well, phage
This is a TWiM bridge right here.
It's just great how you can leverage what people said years ago, why do we have to study how phages lyse their bacteria? Well, now we see, right? Because you can use it. Who knows what the benefits are going to be, right?
Yeah, exactly.
Well, this is what basic science is all about, right? Like there's a reason to study exactly how things function because we can apply them in tons of different ways.
So the movies that Jeff showed, they had a fluorescent reporter, and you see starting from one cell, all of a sudden you have this massive fluorescence, and then it goes away as they all lyse. Right?
Yep. And they leave behind what's called the ghost shells of the bacteria, which is, pure.
Ghost in the Shell. Did you see that movie?
Yeah. Long time ago. Yeah.
You never saw it?
No.
Oh, you should. It's really amazing.
I call them ghosts of bacteria past, but ghost in the shell.
It could be a title, Ghost bacteria. Those are the membranes or the cell walls left behind, and the contents are out, right?
Yeah. Yeah. So, I think another important thing is that only 90% of them lyse. Yeah. So then 10% of them actually grow back and seed the colony again. So you get this repeated behavior of growing and lysing. And so you can imagine them being in the tumor, growing and lysing and releasing this therapeutic.
And in culture in the lab, will they do this indefinitely, this growing and lysing, or does there come a point when they get tired?
Yeah. So in culture, they will actually run out of nutrients in just a standard culture. But if you use a microfluidic device that could constantly feed them and wash out some of the AHL, you could see repeated oscillations. I think for at least, I don't know what the number is, but at least 20 to 30 oscillations people have seen.
How long is that?
In vitro, in culture systems, it's usually a few hours, like 2 to 4 hours, something like that.
But in theory, you could go on forever, right?
Yeah.
Now, when you were designing these, did you ever worry that it would go out of control? Like if you were ultimately doing therapeutics in a person, what could go wrong that would make it go forever and ever?
Yeah, I think that's a concern when you're thinking about these circuits therapeutically is how do you combat evolution, and could it, get the better of you, or could it evolve into something more dangerous? I think it is possible. It's very interesting with these circuits. We actually see them to be very robust. So in some studies we've done in mice, they last in the tumors for up to 2 weeks, and they're still lysing even at 2 weeks. We'll take out the bacteria from the tumor and plate them on a petri dish, and all 40 out of 40 colonies seem to still lyse.
It's amazing that's so stable.
Yeah.
And just to plug kind of an up and coming story, ongoing work in Tal's lab is also looking at ways to really make it even more stable. And in addition to, coming up with new targets, really different ways that, like, what is the minimal system? How many copies do you need to? So that's keep your eyes out.
Could you put a kill switch in that if something goes wrong, you could somehow trigger it?
Definitely. Yeah. I think you can have antibiotics as sort of the fail safe, but you could also engineer a kill switch that will, potentially make it stop completely after 20 cycles, or you can trigger it with an inducer or light or something like that.
So that's the synchronized lysis circuit, so the bacteria. And we'll talk about what you put in them in a moment, but let's go back to this idea of using bacteria. So that you say in your paper, this is something that William Coli observed, tumor clearance in patients receiving injections of bacteria. How many years ago was this?
Over 100.
Over 100.
Yeah.
Wow. That's the way these
the pendulum swings back and forth. And,
same thing with, I'm sure you know that people are exploring using viruses for oncotherapy. And it began when people noticed that when people got virus infections, their tumors went away or got smaller or whatever many years ago. And you said bacteria grow in tumors because they're immunosuppressed?
We don't exactly, this is the understanding right now, but that's kind of mechanistically something that Tal and I are exploring more deeply. There is a bit of hand waving to say what makes them exclusively grow there, but we have a few different steps mechanistically that we're testing, because really understanding exactly how that works will allow us to even make it more efficient.
Sure. Well, it's the same idea with viruses, right?
Yeah, why, right?
We think that tumors have poor innate responses, but not everyone does.
Right? I agree with, it's kind of a black box still. There's there's a lot of really good suggestions, but none of them have really been tested empirically. So we'd like to do that.
We started working on a viral oncotherapy project with Seneca Valley virus, which is an animal virus, non-human animal virus that infects people, but you don't get sick. But if you have tumors, it preferentially grows in some tumors, but it's very sensitive to interferon. So if the tumor, and people have published this, if the tumor makes interferon, the virus will not grow. And so people are trying to figure out how to engineer the virus to get around innate responses, like maybe administering it with compounds that blunt the interferon response. But I mean, the viral oncotherapy has gone on for many generations. So I guess this idea for bacteria and tumors is relatively new now, the modern engineering phase, right?
Yeah, I would say in synthetic biology and the engineering of bacteria is relatively new, but this idea that bacteria colonize tumors has been known for many decades. And I mean, I guess in comparison to the viruses, one of the differences is that it's not exactly dependent on the genetics, like the innate immune system, but rather it being a solid tumor and having a necrotic hypoxic core that leads to this immunoprivileged environment. So we think it could potentially be applied to, all kinds of solid tumors.
So it has to be a solid tumor, is that right? I was going to ask you, does it work for leukemias? No.
So I think it would work best for solid tumors.
Okay.
We've had a number of people ask us about that. Just if you think about the fact that, this system requires quorum sensing and lysis, you need a confined space for these things to be triggered.
Yes.
So that's how we view it. We do think that solid would only provide that kind of environment.
That's a lot.
But there's a lot of tumors.
That's 90% of tumors, I think, are solid tumors. Yeah.
So then the idea was, for this paper, you were going to produce a therapeutic, and tell us about what that is.
Yeah. So what we've done here is we're producing a therapeutic that blocks a receptor on the surface of the tumor cells called CD47. So it's a ligand that is normally expressed on healthy red blood cells. It engages a cognate receptor on phagocytic cells like macrophages. And prevents them. It's a don't eat me signal. So normally healthy red blood cells have high levels of CD47 on their surface, and that prevents them from being eaten. As they age, they downregulate CD47. That no longer allows that don't eat me signal to be present, and that causes them to be eaten. Well, it turns out the tumor cells co-opt this signal. They overexpress CD47, thereby preventing their uptake. By preventing their uptake, then they prevent those phagocytic cells from presenting pieces of tumor on their surface. And this presentation of pieces of tumor on the surface of a phagocytic cell is what ultimately ends up priming a T cell that recognizes unique pieces of a tumor cell. So this is where it gets a little bit complicated and deep into the immunology, but if what we can understand is that blocking CD47 will allow greater phagocytosis or more eating of the tumor cells, then we can understand that that will allow the phagocytic cells to present more pieces of tumor and have a better chance of stimulating the anti-tumor T cell response.
And the T cells are these CD8s and CD4s?
So we do see CD4s and CD8s being primed. For the aficionados, it would make more sense that we can imagine how CD4s would be primarily primed through this mechanism, but there is an amount of cross-presentation that's also occurring that does seem to stimulate CD8s. And we can certainly detect that even in an antigen-specific manner, which is actually super cool. One of my favorite experiments in the paper. So we believe that we're priming the T cells. We're having more efficient priming of the T cells. And what's great about priming a T cell response is that T cells will go on and do what they do. They'll circulate around the body. And if they detect a piece of tumor that is, similar to what they initially recognized, then they'll operate again and they'll continue to kill. And it's in a very specific manner. It's in a very highly antigen specific manner. And so you won't have just, broad killing. You'll have very specific anti-tumor killing systemically.
So here's my limited immunology. See, the CD4s will produce cytokines.
Yes.
And some
CD8s will also produce cytokines.
But the CD8s in addition will lyse.
They're also quite cytotoxic on their own. Exactly.
And so the CD4 cytokines, some of those help the CD8s mature, right?
They also help the CD8s.
But they also will lyse tumor cells directly?
There's probably a limited direct cytotoxicity of the CD4s, but certainly having their response is beneficial.
Okay.
What we did not do in this paper, which is commonly done, is depleting just the CD4s or depleting just the CD8s. Those are clearly very important experiments, but for our purposes, we were more interested in knowing whether we had a therapeutic effect.
Sure.
We weren't going to get into the minutia of the who's more important.
I presume you'll do that. It's an ongoing. Yeah. All right. So the CD47, is this on a lot of tumor types, or?
It is actually upregulated on a lot of different human tumors. And you can see that levels of CD47 on the human tumor cells is much greater than on healthy tissue. And furthermore, levels of CD47 or higher levels of CD47 have been linked to worse survival or worse progression.
Well, it makes sense that tumors would be selected that have this. It avoids phagocytosis, right?
Exactly.
So then the question is, why haven't companies 20 years ago made antibodies against this? Or have they, and they don't work?
So they absolutely have. There's not a lot of what we've done here that isn't already standing on the shoulders of giants. Just to make that very clear. These things have been discovered. We've just put them together in a very novel way in really incorporating technology in a way that's a bit different, but, quite effective. So it has been shown. There have been clinical trials using an antibody against CD47. But if you remember, as I mentioned, CD47 is highly expressed on red blood cells, right? Healthy red blood cells. How we deliver antibodies in a human scenario is often through intravenous infusion.
Yeah, right.
So if you consider the relative antigen sink, or, where would the target molecule be highest? Well, in fact, what you see when you deliver it intravenously into humans is you'll see an acute thrombocytopenia and also an acute anemia. What this means is that the first CD47 molecules that the antibody you're injecting will be on the red blood cells.
Right.
And so to get effective concentrations of this antibody into the tumor tissue is quite hard when you basically have this, wall of CD47 expressed in the blood. So, and on top of that, you see these, acute thrombocytopenia, acute anemia, consistent with really the antibody working predominantly on the red blood cells that it encounters first. Sure. So what our system allows is for this nanobody that actually binds with much higher affinity than the antibody to CD47. It's made locally. It's made within the tumor tissue. So it really avoids this anemia. And we are able to show that in the paper as well, that we don't see anemia as we would with the traditional treatment.
All right, so that's the unique part of this paper. You decided to have your synchronized lysis E. coli produce this nanobody. So let's go into that a little bit. First, why a nanobody? What is a nanobody?
So a nanobody is super cool. So if you're familiar with an antibody, then it's made up of heterodimeric chains, two heterodimeric chains, which is a light chain and a heavy chain that come together through disulfide bonding. So you end up with really 4 polypeptides that make this divalent antibody molecule. Well, it turns out, in fact, that other animals don't, such as llamas and alpacas.
Camels, right? Camelids.
Yes, exactly. So they only rely on heavy chain only antibodies. And in fact, we can use that technology to try and create antibodies against one particular molecule of interest using, in fact, phage display. And so this was done by Chris Garcia's group, in addition to Hitaplua. This is a collaboration between their groups and the Dugan Laboratory. And what they did was they discovered a nanobody that targets CD47. And what's nice about it is it's only 15 kilodaltons, approximately. And it's one polypeptide chain that folds on its own and gives you essentially the binding dynamics that you would see for an antibody. An antibody being 150 kilodaltons, right? So if you can imagine a bacterium expressing just this small nanobody, you can get tons and tons of the effective protein made. Now, the problem with nanobodies, I'm making it sound like it's just like the perfect thing. Why aren't we using this all the time? Well, the problem is that their stability is quite low. But again, because of the advantage of our system where we're locally producing it, kind of like this intratumoral bioreactor is what we're using the bacteria as, the half-life isn't as much of an issue because it's constantly and synchronously being delivered.
Okay. So, Tal, you engineered the gene for this nanobody into your SLC E. coli, right?
Yeah, that's right. So Shreyan, who was the student, actually did this. He did it, in an interesting way because he took the original circuitry, but he actually split it up into two plasmids. So one was just encoding the lysis circuit, and that would allow the bacteria to have this function. And then the other one is a high copy plasmid so that you can make a lot of this therapeutic nanobody, as much as we could possibly hope for. And then that new system that we built is the one that we tested therapeutically in mice.
And so you first showed that these bacteria make the antibody and it's synchronized production, right?
Yep.
You did some studies with cancer cells in culture, right?
Yeah. So we purified the nanobody. We showed that we could express it, get the right size. We showed some increased phagocytosis when you applied the bacteria lysate that makes the nanobody. And those were some of the preliminary in vitro experiments that we did.
You have these A20 tumor cells. These are lymphomas, I guess. Right? They have CD47. So you show your nanobody binds to those. It stimulates phagocytosis. And for that, you used macrophages from mouse bone marrow, right?
Yeah, bone marrow derived macrophages. They're pretty
easy to get.
They're pretty easy to make. I have tons frozen down. We tested just a bunch of different
and you
lengths.
can show that the nanobody stimulates the uptake, right?
Exactly.
Of the tumor cells by the macrophages. So that's all kind of preclinical, right? Well, preclinical and
preclinical going into
before mice.
Yeah.
Because actually mice are preclinical.
We call that pre in vivo, I guess.
Pre in vivo, yeah. So then you want to go into mice. And what did you do? Tell us about those experiments.
So actually for mostly for simplicity reasons and just to have the easiest system, Shreyan was doing intratumoral injections of these mice with the bacterial system. That just allowed us to, avoid the complications of systemically delivering the bacteria and then waiting for to target. We did that later, but he would do about 4 intratumoral injections over the first 10 days with PBS and with bacteria that have the lysis circuit but don't express the therapeutic or the lysis circuit with the CD47 nanobody therapeutic. And what he saw was that when the bacteria express the CD47 nanobody, you can see these A20 lymphomas completely go away.
So you establish, so these are mice where you inject tumor cells initially, right?
Yep.
In this case, it's a lymphoma, A20, the same one you used in vitro to look at the efficacy of the nanobody. Are these nude mice?
No, these are mice that are
completely competent,
fully immunocompetent.
So why do they grow a tumor then?
Well, it's because there are orthotopic lines that essentially they're from the BALB-C background.
Oh, I see.
So they go undetected, essentially. There's not enough mutations for them to be recognized as foreign.
So they're wild type, they're BALB-C mice. You inject the cells, and then they grow into tumors. And then you put in your bacteria, as you said.
Yeah, that's right.
Right into the tumor, right? It's easy to see because the tumor's a big mass, right?
Yeah, and we inject them subcutaneously.
Yeah, and we wait for them to be about 100 millimeters cubed or 100 to 150 or so.
Okay, and what do you see when you have this SLC with the nanobody in these tumors?
And we see the tumors completely resorb, which is actually was pretty surprising.
It's impressive.
And completely gone.
Yeah.
Yeah, and there are pictures
probably
of
open
that.
them up and look, and you don't see anything, right?
Yeah, it's just...
It's gone. Wow.
Yeah.
And you say the mice, if you don't do anything, do these mice die?
Yeah.
Eventually they will die, right?
They even die with the bacterial therapy that doesn't express CD47 antibody.
Okay.
So you'll get kind of an initial nice boost, like maybe a small decrease in growth just due to the bacterial ligands stimulating an innate immune response. But in terms of eventually clearing out the tumors completely, it requires that the bacteria also express the CD47 blocking nanobody.
And you also say that, all right, so these surviving mice, you re-challenge them with tumor cells.
Yeah.
The tumor doesn't grow.
Doesn't grow.
It's amazing.
It's kind of a hint there, right?
Yeah, there's some memory, right?
Some memory immune response
right.
that's...
Do you think this would last the lifetime of the mouse? I don't know if you've tried that. Because here you challenge them, a couple of weeks later?
We 90 days later, right?
Yeah.
Something like that's a long time in the lifetime of a mouse. I don't know if it would last the entire lifetime. My feeling is probably a good amount of time. Yeah, it's a memory T cell response. The recall should be around for quite a while.
Of course, that is in mice, and eventually you'll have to see in people what happens. And then you'll only know by looking to see. It's like a vaccine. You see, vaccinate people. You see how long a memory lasts. Mm-hmm. It can be good or it can be crappy, like flu vaccine, where it lasts less than a year. Right.
Or sometimes doesn't even work.
Yeah, it may not even work. Sure. Then you tried intravenous administration, right? Mm-hmm. And why did you do that?
The intravenous administration was really just to highlight the fact that the bacteria can target the tumors selectively. And we did some experiments that show after a few days and after a few weeks, they're only in the tumor and they're not in the healthy spleen or kidney or liver. That's
remarkable. I remember you talking about that at your seminar. You can intravenously inoculate a lot of bacteria, right?
Yeah.
Mm-hmm. 10 to the 10th, is that the right number?
Oh, I don't know if we've got, probably that would work, but we do normally to the 7th.
10 to the 6th. I see, there it is. And you only find them in the tumors.
Yeah.
Nowhere else.
I know.
It's remarkable. It's E. coli. Right? You would think.
This is not that surprising, actually, because when I was in graduate school, we were doing an experiment to test something just completely unrelated that came out in the literature, and we were injecting tons and tons of bacteria intraperitoneally and testing 2 to 3 days later, and you can't detect any, you can't find them. Just 2 to 3 days later, no matter how many you injected, we really couldn't find any in any of the healthy organs.
Is that a particular property of the E. coli you're using, or would other bacteria do it as well?
People have shown it for a couple of types of bacteria. Salmonella also behaves that way, and Clostridium, and also some other obligate or facultative anaerobes that can survive the hypoxic tumor environment.
Because I remember when Jeff Hasty was here, he was using a probiotic E. coli. He said you can buy it from Amazon, right?
Yeah, E. coli Nissel, 1917.
Right? Yeah. So it's E. coli still, but it's just a particular strain of.
Is that what you used here, Nissel, or is it different?
Not in this one. We used a non-pathogenic E. coli that's more used for cloning.
Now, the other issue, you showed different kinds of tumors could be treated, right? What other ones?
So breast as well as melanoma.
Okay.
And that actually the melanoma is performed in a different background of mice in the C57 Black 6 background. And so we certainly see efficacy in those two models. It's a bit different. They are quite aggressive models, but we still do observe efficacy. It's just, albeit maybe less.
And then you looked at metastases as well, right? So does the A20 metastasize at all?
It certainly does.
Yeah. So you inject it, you get a mass, but then you can find it in other tissues. And the others, the breast cancer and the melanoma, they metastasize also, right?
They do. At different stages and depending on the breast certainly metastasizes quite well. I believe that was to the lung. The A20 we were monitoring, the lymphoma we were monitoring metastases to the liver. And what we could note is that when we treated with the therapeutic strain that expresses the CD47 nanobody, again, we saw reduced growth of the primary tumor lesion, and we also observed fewer metastases. So it's a bit difficult to know whether the reason we're seeing fewer metastases is because the primary mass is smaller. But regardless, it is quite interesting to also observe a reduction in the number and size of metastases.
So you can inject into the tumor mass, and this will reduce the number of metastases that you see. And I guess that's related to survival because you said in the original experiment, 80% of the mice survived over 90 days. So some did die, right? Maybe that's related to metastasis.
Oh, you mean some did die?
No, you said 80%.
We didn't have 100% of them survive. So
one died.
It could be an injection problem. That's probably the most likely.
Well, basically, for what you can see is for multiple tumor types, you can regress the original tumor plus metastases with this injection.
Yeah.
And so that's the interesting part here. The metastases, obviously, you're not injecting the bacteria into, but they're being taken care of in some way.
Yeah, right? And so that's really what we'd like to harness moving forward. And actually, in these later experiments, what we show is that we believe that's because we're priming an anti-tumor immune response that's T-cell mediated.
Right. So then actually the next series of experiments have to do with that, right?
Yeah.
You're priming anti-tumor T cells. So the idea is that phagocytosis that you're stimulating by having this antibody is allowing uptake of tumor cells, and then the APCs, the antigen presenting cells, are presenting those, and now you're getting a nice T cell response.
Exactly.
Right?
And so you're locally priming a T cell response, and the question is that locally primed T cell response effective at treating or killing off tumors in other locations?
Right. And we'll talk about that. First, I want to ask you, you have this section where you're talking about antigen presentation, and you talk about a protein called SIRP alpha. I don't know. What is that? I have
idea. So I kind of touched on it before, but it's SIRP alpha. SIRP alpha is present on the surface of the phagocytes.
Okay.
And when CD47 engages with SIRP alpha, that's what transmits this "don't eat me" signal.
I see.
Yeah.
I see. Got it.
So signaling through SIRP alpha would essentially block phagocytosis of the tumor cells.
And you were able to show that the T cells that result in culture, they recognize specifically the A20 cells in culture, right?
Yeah,
so they're antigen specific, right?
If we treat with irradiated tumor cells, we see that the majority of the T cells that were primed during the initial treatment event do respond to irradiated tumor cells and produce interferon gamma.
Got it. So you getting T cells, which, as you said just before, suggested should have a systemic effect. Right?
Yeah.
So then that's the next series of experiments which address that. Why don't you tell us about those?
Yeah, so what we did in this scenario is we injected tumor cells subcutaneously on both flanks, so both the right and left flank of the animal. But then because we were treating intratumorally, what we did was we only delivered the treatment into one flank, so let's say only the right side. And as we expected, we saw that the tumors that were injected, began to shrink with the same kinetics that we saw in the other model. But then at later time points, consistent with the priming of an adaptive immune response, specifically a T cell specific response, even though the other tumors on the opposite flank that were never treated, we started to see that those were shrinking as well. What this indicated to us was that T cells that were specific to the tumor cells were being primed in the treated location, and that these T cells could then traffic over to the untreated tumor and in fact, kill off those tumor cells as well.
And how do you know the bacteria aren't simply going from one tumor to the other?
So we did that experiment as well. And we did it in two ways. One is we used luminescent bacteria that you could track using an IVIS in a live mouse. So we injected bacteria into one tumor, and we looked at the luminescence in that tumor. And then we tracked over time. We didn't see any luminescence show up in the other tumor. And then we also eventually took out these tumors, both of them, homogenized them, and plated it on Petri dishes. And we only see bacteria grow in the ones that we injected, in the tumors that we injected.
And this is what you call an abscopal effect, which is a word I hadn't heard of until we started working on viral oncotherapy, because the same goal is to get an abscopal effect where you inject the virus into one tumor and the other one goes away, not because the virus is going there, but because of immune responses.
And this is a term borrowed from radiation therapy, where the radiologist will see that they will irradiate one tumor mass and then observe that other tumors that were never irradiated are also shrinking. And so we are borrowing this term from radiation oncology.
Right. Now, your idea is that the injection of the bacteria into the tumor is leading to a tumor-specific T cell response, and then those T cells are circulating. Do you have to, at some point, prove that?
So we are able to detect that in the untreated tumors, when we isolate T cells, that we can detect tumor antigen-specific T cells within the untreated tumors, which would indicate that, in fact, the anti-tumor-specific T cells that were primed in the opposite tumor have trafficked over to the untreated tumor and are at least participating in that effect. We, of course, do want to pursue these kind of mechanistic experiments further, not just looking at, a couple of antigens, but more broadly, just really the specificity of all of the T cells that are there. But I think we were able to, kind of show that pretty solidly that is what's occurring.
And that's this peptide that you have in the paper, which, DYWGQGTEL, which is a tumor-specific peptide.
It's a tumor-specific peptide corresponding to the antibody, which is uniquely expressed by the lymphoma cell itself.
And you can show that you've got T cells in these mice that recognize that
exactly,
peptide.
Yeah.
Because when you add the peptide, the T cells, they make interferon gamma, right? Is that the assay?
Mm-hmm. So that's my favorite experiment.
Yeah, okay. That's great. So you have an abscopal effect, which is independent of bacteria trafficking. It's so cool.
I know.
I just think that's amazing.
It's really cool.
Let's see. So we have, so the other part is the immunostimulatory products made by bacteria, right? Do you ever look at that directly, or is that just something you think is happening?
We're doing that now. Mm-hmm. We are interested to know what particular TLR or toll-like receptors are being activated, which particular ligands are activated. Also, Tal and I, we've been, kind of throwing back and forth a bunch of different strains of bacteria. And so it is important to know how these different strains activate the innate immune system and whether that correlates really to their overall efficacy in a given, tumor scenario.
Right.
So we are looking at that using some pretty basic techniques.
Are you going to look at knockout mice, for example, for these different pathways?
We have proposed doing that. Depending on what grants come through, there will be things that we pursue more deeply.
I mean, I can imagine doing that for the innate response, right?
We're really interested in doing it just more broadly for this intratumoral homing response. Like when we inject bacteria intravenously, it's easy for us to say, it's the lack of the innate immune suppression within the tumor that's mediating this homing. Yeah. But again, that's quite hand-waving, right? That's a bit of conjecture there. It hasn't been empirically tested. And so there are certain things that we would like to test just quite thoroughly to really assess what is participating in this particular homing event.
And I assume, so you think there may be some specific interactions going on there? So you could look at the bacteria as well. You could look at maybe a transposon mutant library, right? Yeah. To see what bacterial proteins are involved in the homing. That would be cool.
Yeah, just in the homing or in the therapeutic effect.
Yeah. And what about the CD4 versus 8? Would you also use genetically altered mice to look at that, or would you just do that by depletion?
We can do that by antibody-mediated depletion.
It's easier to do by depletion.
But that, again, might change given within the given tumor scenario, depending on what types of tumors we're using. Right now, I think our interests are really to assess the broad applicability of these approaches using additional targets that we've since developed, and also, looking at different tumor models and different delivery methods.
Right. It's funny. I'm looking at the references here. There's a paper in PLOS One. ALX148 blocks CD47 and enhances innate and adaptive anti-tumor immunity with a favorable safety profile. And the first author is Steve Cowder, who was my graduate student.
Is the second author Tracy Quo?
Oh, it's his wife. I know that, right? Let's see.
I know Steve.
Tracy Quo is his wife.
Tracy.
She was a graduate student here.
Yeah, and then she was a postdoc in Mark Schlüssel's lab.
So I know Steve went to Genentech initially after here, but apparently he's now at some small company. Well, I don't know if it's small, but it is called ALX Oncology.
Hi, Steve and Tracy.
Wow, what a small world.
I know. I didn't know that Steve worked with you. That's pretty cool.
He did. Yeah, I'm looking at S.E. Cowder. Can that be Steve? Yeah, it's him apparently. And so this is an example of a company looking at the therapeutic effect of just the monoclonal against.
Yeah, so they did a bunch of non-human primate work there, and I believe they have a version of this particular therapeutic that is maybe different than other ones that are being trialed at the current time. It's hard to find out exactly what they're using.
Right, of course. I want to tell you. All right, so I have a couple of other general questions. So, is this ready to go into people yet, or do you have to do some more things?
Definitely before we go to people, we have to do some, I would say, further preclinical experiments to look at efficacy and check different mouse models. And we want to, make the bacteria more stable. I think Nick alluded to that, is integrating the system into the genome and looking at evolution, like you mentioned, how well does this thing function? And then also toxicology studies we need to do. There's a sort of array of experiments we need to do before we put this into people.
But that's not going to happen in your laboratories, right?
Well, we're trying. We're certainly through the TRX, for example. So through Columbia Technology Ventures and funds provided by the NIH, they help with the development of certain preclinical therapeutics and getting them to a stage where they can at least attract additional funding to complete these toxicology experiments. So we do want to be involved in every aspect of this because we think our motivation to push this forward is truly driven by a desire to help people and less driven by the financial gain associated with it.
Yeah, sure.
And so we want to be involved in every aspect as much as possible. So we're pursuing any avenue that will allow us to do that. And luckily, Columbia provides these types of programs.
You can outsource a lot of this. My wife worked for a company of 8 people that outsourced everything, all preclinical studies leading to phase 1 and 2 is all outsourced. And so that's possible to do that. And you can be very flexible that way. You say, okay, and you can do toxicology because there are companies that specialize in doing that, right? Because it's just a matter of giving something to animals and cutting them up and looking. And
I think there are certainly, some things that to us might just kind of be boring experiments to do, but they're clearly
you have to do it. You have to do it.
So you just throw money at that instead of.
I think it's very exciting that this has therapeutic potential, but obviously there's a lot more you have to do before that. And I just wonder how much people are going to push back. I guess if people are willing to take a virus to cure their tumors, they'll take a bacterium as well, right?
Yeah, and I think a lot of the microbiome research and people taking probiotics and, wanting to get their kids dirty and, not necessarily seeing bacteria as bad has helped this field of, bacteria therapies.
Yeah. Do you ever think of I don't know if you think about viral oncotherapy at all. Obviously it's comparable. You think this might have advantages? Can you envision using both in a patient? Both viral and your approach maybe would be complementary or even synergistic?
Definitely, yeah. And we have some projects in the lab that are trying to look at some oncolytic viruses and comparing them with bacteria and what are the advantages of the two. So I think it's very interesting to look at.
So we work on Seneca Valley virus. If you're interested in trying that, you can do the same kind of assay. You make tumors in mice and you inject it. Right. And the cool thing about SVV is that its receptor is pretty much specific on the tumor microenvironment. So the blood cells that feed tumors have TEM8 it's actually the anthrax receptor, TEM8. It's a receptor for this virus. So it kills those cells and it prevents the tumors from getting the blood flow that they need. And it's not present on other tissues. So you can inject it into tumors in mice and it will shrink them. So if you ever want to look at that in combination, we'd be happy to help you out there.
Yeah, that's really
That'd be
cool.
A cool combo.
Yeah, I think one of the cool things is that, with all the synthetic biology, you could also engineer their interactions.
Yeah.
So somehow the bacteria can help the virus or the virus can help the bacteria, or one can regulate the other in some kind of safety way.
Somebody asked me once, I forgot who it was, if you could. Oh, it must be somebody. It's an undergraduate at Columbia. Asked me if you could engineer a bacterium to make a eukaryotic virus for this purpose. Maybe it was someone, a summer student of yours or something.
It could be, yeah.
You wanted to know the obstacles to, I have the email. I can look it up when we're done. You wanted to know the obstacles to getting, a eukaryotic virus made in bacteria. And there are many obstacles, right? Depending on the particular virus. But with the simpler viruses, all you have to do is make the proteins and the nucleic acid, and they should self-assemble. So that would be interesting to use the bacteria to deliver the oncolytic virus, right?
Definitely. Yeah.
See, these are the kinds of things that, maybe back in the day you would come up with this kind of idea and somebody would be like, that's crazy. Like,
yes,
that's silly.
That's right.
Like, don't even think about that. But, I don't know how many of your viewers are like undergrads or pre-undergrad, but it's a good idea to think a little crazy sometimes, because you get two like really creative people together and sometimes you can make those things happen. So
I totally agree, because I can tell you so many times in my career I have said that's crazy and it turns out to work. So I've stopped calling things crazy. When they first started talking about using viruses to treat tumors, the first one was Riovirus, right? And that was the first company that was made. I thought it was crazy. I said, how can you treat a tumor? Now we can do it. We can modify the virus. We can do all kinds of things that we didn't know before. So you should never say never. I remember they wanted to sequence the human genome. I said, well, what are you going to do that? What a waste of money. Give it to people for R1s. Obviously, it's great, right? You can learn a lot. So if he said to me, I want to make a eukaryotic virus in bacteria, I said, okay, well, it's not easy, but here are the obstacles. But, as long as it doesn't take you 10 years. But even then.
Even if it did.
Axel's postdoc, Linda Buck, took 10 years to identify the odorant receptor.
Seems like got it. It's absolutely worth it.
Totally worth it. But you have to be willing to invest that kind of time in it, and maybe not get anything. So that's a tough one to call. But anyway, the other question I had for you, checkpoint blockade therapy often involves monoclonals. Can you improve it by using the antibodies in your bacteria?
I think that's definitely a good idea. We actually have a paper together that's on bioRxiv from a student that's in our lab, Candace, who has made PD-L1 antibody and a CTLA-4 antibody. And we see some pretty good therapeutic results there. And it's actually mirrors the clinical scenario where when you combine the two together, you see a much stronger effect.
Yeah. And plus, if I'm not mistaken, the checkpoint is by intravenous.
Exactly.
Right?
Well, in the clinical scenario.
Yeah. And so your approach may be better.
So local delivery in this case seems to work quite well. And I think the other cool things in Candace's paper are really these optimizations to integrating the synchronized lysis circuit, etc.
Yeah, because it makes sense that your two approaches would synergize, because you're making T cells, and if they get exhausted, they're not going to work. So the checkpoint blockade could take care of that, right?
Yeah. And so in the case of the CD47, by the way, we call that kind of an innate checkpoint.
Okay.
So that's like innate checkpoint blockade. But in terms of your canonical, your more broadly spoken about checkpoints, PD-L1 or PD-1, CTLA-4. Yeah, we are coming up with clever ways to maybe have the bacteria sense when a conversion has occurred in the environment, maybe sense when exhaustion is imminent,
right,
and then start to produce a different nanobody that would target that particular response.
Got it.
So we really do want, to kind of do synthetic immunology in the sense where we can sense the environment and then respond and provide a particular treatment depending on what has been sensed. That's the dream there. But, we're doing what we can.
You don't need to get there right now. I mean, you could get this into people first, as is, or almost.
Oh, absolutely.
Right,
I think
that save that for the future.
Absolutely.
Now, you said the last sentence of the paper, you say, owing to the observed abscopal effect, we provide a future strategy for treating metastatic lesions through the injection of accessible primary tumors. So that is the key here. You need a primary tumor that's accessible, right? So certain brain tumors could be accessible, right? They do inject. Did you know that you can inject poliovirus into glioblastomas and get some effect? But not every tumor is going to be accessible, right?
Not every tumor is going to be accessible. That's correct. But again, these are the reasons that we're also looking at the intravenous delivery approaches.
Yeah.
And, potentially oral delivery approaches that can colonize lesions in the intestines and potentially also in the liver. Common sites of metastasis being liver metastases. If we can prime in a metastasis and then have the T cells circulate around and kill off other metastases and primary lesions, that would be an awesome benefit, I think, to patients and just really, pushing the field forward in that regard would be pretty cool. But yes, there are ways to target lesions surgically. We prefer to, hopefully be able to access lesions either intravenously or orally.
I can see that happening. I just read a paper, this amazing paper, where they did phage therapy of a guy with a infected aortic implant. So, you have sometimes you have to replace the aorta with a piece of Dacron, and he's got infected. And he actually had an open wound on his chest. But they went in and just poured the phage on his implant. It was so covered with scar tissue, they couldn't inject it. They just poured it on, and he was cured.
Wow.
It lysed all the bacteria. Right. I think it was a Pseudomonas infection. They wanted to open up and go in, but they said it was so covered with scar tissue, they were worried. So they just poured it, the tube of phage, they poured it on the Dacron, and the guy recovered and went home. The sad part is, the day the paper was published, he died that day from a heart attack or some other.
Oh, it's not related.
But this is a remarkable story of how phage therapy is the other part of this that is amazing. It's just going forward in ways that could never, because you can do some engineering. And in the past month, there have been 2 cool phage papers out which show that you can do things differently. You just don't take a phage that kills the bacteria and give it to people. You can do clever things like have the phage attached to an efflux pump so that if the pump changes to prevent the phage from binding, the bacteria become antibiotic sensitive because they can no longer pump out the antibiotic, right? Strategies like that. It's I would include it with this engineering that you're doing. It's the same idea.
Yeah, we've been thinking of some crazy phage mid type things, just making multi-level triple organism. Well, I guess it depends if you call it phage an organism. But we've been thinking of some ways of integrating, host, bacteria, phage.
Yeah. The system you have of using bacteria to deliver things can accommodate a lot of things, as you know. I'm sure you think about it all the time. Not just drugs, but biologics, viruses, and maybe bacteriophages, especially if you can inject and the bacteria go to where you want. So both of you have other research programs in your labs, right? So Nick, I know you do immunology, and this is a kind of a side project, right?
Yeah, I'd say it's about what a third of my lab works on. And, everybody in the lab is aware of these parts and, thinks of ways of integrating these clever methods into their projects, whether related to tumor immunology or not. I
mean, this could totally become your entire lab, right? From what you've said today, there are a lot of cool things ongoing.
It certainly could. But, at the heart of it, I am a basic immunologist. And as we discussed earlier in this podcast, I think, there's a true benefit to studying basic science.
Of course.
So this is certainly our most therapeutic minded, directly and, clear, like therapeutically focused approaches. But the more we learn about the basic operations of the immune system, the more we'll be able to integrate new systems into this platform and make it even better.
Yeah. Tal, what fraction of your lab is this project?
I'd say the lab is mainly focused on synthetic biology and bacteria cancer therapies. So probably 80% of our lab is working on this, whether it's different therapies or different circuits or different strategies or different organisms. We have some very basic bacteria pattern formation studies, and we're interested in that, how bacteria make patterns on Petri dishes and how they cooperate and how we can engineer them and what could they be used for. But we're pretty committed to the bacteria cancer therapy space.
All right. One last thing. Is there anything we forgot to tell our listeners that you think is important with respect to this story? Something we didn't cover before we stopped? Did we tell the story well?
I thought it was really well.
I think we did a great job. I think you led it along quite well. I guess.
Okay. I just want to make sure, because sometimes when it's not my field, I just want to make sure that we covered it all and that people will understand. But of course, people do send in their questions. That's the cool thing about our podcast. We have a wonderful audience who, if they don't understand, they'll email us.
Yes, send them in.
Cool. And we'll send them to you and you can answer.
Absolutely.
And so this is a lovely story. I encourage everyone to check it. It's probably not an open access paper, though, unfortunately. Nature Medicine, right?
It might be currently for a little while.
But as always, I always tell people, if you want a copy, just email the authors. That's
we were provided with a link for free access
for a while, so you can always do that. And it's a lovely story. I think this is great. I look forward to hearing more, and I look forward to you guys coming back and talking about the future of this. It would be really cool. All right. This is TWiM. You can find TWiM on any podcast player. Most people listen on a phone or tablet. Please, all we ask is that you subscribe. You push the subscribe button so you get every episode. And we know how many people are listening. That really helps us to push the shows forward. So you can do that on any podcast player. We have all of our notes, show notes, etc. at microbe.tv/TWiM. If you really like what we do, consider supporting us financially. You can go to microbe.tv/contribute, and there you could use either Patreon or PayPal and say, give us $1 a month. And that would be for you not much. It's not even a cup of coffee. Here in New York, you can't get anything for $1. Nothing. So you might as well give it to us. We produce 5 really good science podcasts. And all it is $1 a month. And it helps us to be able to do things like travel and do more podcasting and so forth. So microbe.tv/contribute. And of course, if you have questions or comments about what we talked about today, TWiM at microbe.tv. My guest today, this is an all Columbia TWiM. I think it's the first ever.
Woo-hoo.
They're all here at Columbia University. Nick Arpaia is at the medical school here at Columbia. Thanks, Nick.
Thank you.
Good to have you. Good luck with this lovely story. I really look forward to see where it's going. Tal Danino's at Columbia University. Thanks for coming up.
Thank you.
It's a pleasure. And I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank ASM for their support of TWiM and Ronald Jenkins for the music, ronaldjenkins.com. This episode of TWiM was edited by Ray Ortega. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.